All right, we're going to finish talking about the location of these various pumps and channels along the neuron. So a little anatomy of our neuron that we need to understand before we talk about the physiology. In the last video, we finished talking about the location of the chemically gated, where you can find those chemically gated ion channels for sodium, potassium, and chloride. So the next type of channels that you need to know where their location is the voltage gated channels. And those are voltage gated sodium channels and voltage gated potassium channels. So where you can find those, kind of scooch down a little bit here, you can find them in the, they call this the initial segment of the axon, also the axon hillock. So you can find them here. And then you can find them, they call this the conductive segment. The axon is the conductive segment because it's actually conducting the nerve impulse all the way down the length of the axon. So this is the place, initial segment and then conductive segment or down the axon, where you will find the voltage-gated sodium and potassium channels. All right, now we are down to the last part of our neuron, which includes these little axon terminals, or they also call them synaptic bulbs. They call that part of the neuron the transmissive segment, and that's because those are the parts of the neuron where we're going to get the release of a neurotransmitter that'll be transmitted to the next cell, whether it's another neuron or a muscle cell or a gland cell. So they called this part of the neuron the transmissive segment. So here in the axon terminals, or the synaptic bulbs, you will find voltage-gated calcium channels, and we talked about those when we talked about muscle contraction. So when we talked back in chapter 10 about how the reason the neurotransmitter gets released from the little synaptic vesicles from the axon terminals is because the action potential that makes its way to the axon terminal will trigger the opening of calcium channels, and those little calcium channels open. Calcium will flow into the axon terminal and trigger those synaptic vesicles to release their neurotransmitter. So we didn't call them voltage-gated calcium channels, we just called them calcium channels, but they were actually voltage-gated ones. So they opened because there was a change in the membrane potential or voltage across the plasma membrane at the axon terminals. So now and so calcium is higher outside the cell than inside the cell. So we also have these calcium pump channels that are also located. And so what they want to do is keep the calcium higher outside the cell than inside the cell. So that way, whenever we get a signal action potential that makes its way down to the axon terminals, then these voltage-gated calcium channels open, calcium will flow in and trigger the synaptic vesicles to release their neurotransmitter. So we have little calcium pumps that will continually pump calcium out of the axon terminal to keep the concentration of it higher outside the synaptic bulb or axon terminal than inside. So that's the type of channels that you can find in the transmissive segment, which is the axon terminal or synaptic bulb area of a neuron. All right, so again, this is a little bit of anatomy that you should spend some time learning before you start going into the information about the physiology, because it's these pumps and channels along the neuron that are playing a role in the neurophysiology of a neuron. So they're playing the big role in it. So it's important to remember what they do, where they're located, so that you can understand the neurophysiology better. All right. Okay, so I'm going to head to our objectives because I didn't do that before. But so in the last video and then this video, we talked about the pumps and channels, and we talked about their location, and we talked about the different neuron segments. So we talked about the receptive end and the conductive end and the transmissive end. So it's important to know what role each part of the neuron plays in the functioning. So the next thing that we're going to talk about is some terms that we're going to use when we talk about neurophysiology. And then the next video will jump into how the neuron functions, so the neurophysiology part. All right, so let's go back to our textbook, and I think it's on the next page. Okay, so we are going to need to understand a little bit about electricity before we start talking about neurophysiology. So how neurons work and how they send signals and communicate is an electrical event, and it's caused by electrical energy. So what we need to understand, I'm going to go down to our picture here of a neuron, just kind of, so we have our cell body, and then you can see a couple of dendrites, and then the axon, and I guess that's representing the axon terminal. So what we need to, let me see if I can make that bigger. Yeah. So what we want to understand is that there are two areas associated with the plasma membrane of a neuron. So here's our plasma membrane of a neuron. They're kind of focusing on the axon part. But there's two areas. There's outside the plasma membrane, and then there's inside the plasma membrane. So those are our two areas, and they are different. And how they're different is that they don't show it all, but on the outside of the plasma membrane of a neuron, there are a bunch of chemical substances like ions and molecules. So they're all out here, and some of them will have a charge. So some of them will have a positive charge or a negative charge. So we talked about, ions and how they get a charge. So we will have some that are positive and some that are negatively charged. And if we count up all the positively charged ions and all the negatively charged ions, or molecules too, it doesn't necessarily just have to be ions, outside the plasma membrane of a neuron, we will notice that we have much more positively charged ions or molecules on the outside of the plasma membrane of a neuron than we do negatively charged ones. So what that does is give us an overall positive charge on the outside of our plasma membrane. Now, if we look on the inside, it's a little bit different. So if we look at all of the chemical substances, ions, and molecules that are on the inside of the plasma membrane of a neuron, we'll notice that we have a lot more negatively charged ions and molecules on the inside compared to the positively charged ones. And so what that means is we have an overall or a net negative charge on the inside of the plasma membrane of a neuron. So what we end up with is a charge difference between these two areas. So there's a difference in charges between these two areas. And so we can actually measure that. And we measure it in millivolts. And so that is what the term voltage means. Is just that's what we use to measure the amount of difference in electrical charges between two areas. So that's what voltage is. So when we talk about a neuron, we'll talk about this amount of difference in electrical charges, usually using a value. So we'll have like a value that we'll talk about when we talk about the difference in those charges across that plasma membrane. All right. So having that charge difference across the plasma membrane kind of sets the stage for movement of charged particles across the plasma membrane. So if you remember in the last video, we talked about the various membrane proteins, ions, and channels that are present in different areas of our neuron. Whenever those open, then we can start to get the movement of charged particles across that plasma membrane. And we can actually change this voltage. So if we start getting like positive, in this case the sodium's positive, so if a bunch of positive sodium ions start coming into the area inside the neuron, then we can start changing this amount of difference in electrical charges. So, and then if, potassium is a positive charge and it starts leaving the cell, again, we can change that amount of difference in electrical charges in between those two areas. So anytime we have charged particles that are moving across a plasma membrane between two different areas, we call it a current. So that would be considered an electrical current because it's charged particles that are moving across that plasma membrane. All right. Oh, I almost forgot about resistance. So resistance is what will prevent or oppose the movement of those charged particles. So obviously the plasma membrane prevents the movement of charged particles whenever, just with it being present there. So, and if a membrane protein is closed, let's say this channel is closed, and the ion can't move through there, that's causing resistance to the movement of those charged particles. So that's something that will kind of oppose the current or flow of electrical charges. All right, so those are some key terms to be familiar with, and that's kind of an overview of how the neuron is going to communicate or send a nerve impulse. It's all based on the changing of this voltage that's the difference in these charged particles across the plasma membrane. So anytime we change it, which we change it by the opening and closing of membrane proteins, whether they're channels, most of them are channels, then we'll get a change in that voltage, and that will be our electrical event. So let's go back up here, and so this is the area of the textbook that talks about the terms that we just looked at in that picture. So voltage, current, and resistance. So we should be familiar with what we mean when we're talking about the concept of voltage and current and resistance when we're talking about neuron function.